This new unit is on the cell cycle, mitosis, and meiosis. And so this first set of notes is going to cover the cell cycle in general, and then specifically the process of mitosis. The cell cycle refers to the life of a cell. And what this is showing us is that this cycle continues over and over. So the cell will go through these different stages, it will eventually divide, and then it will re-enter the cycle and continue doing this throughout its lifespan. Cell cycle is a continuous series of events that take place in a somatic cell. What happens during the cell cycle is that the cell will grow, it will duplicate or copy its DNA, and then there'll be cell division to produce two daughter cells. On average, our body is doing this and making about 2 trillion new cells daily. That is to replace old or worn out cells, it's to repair injuries, and if you are still growing, it's actually to help your body grow. So we said in the definition that the cell cycle and mitosis has to do with the somatic cell. A somatic cell is just a fancy name for a regular body cell. All of our somatic or regular body cells are diploid, and diploid is abbreviated to N. Diploid means that the cell has a full set of chromosomes. It has all the chromosomes that it's supposed to have. In terms of us as humans, we have 46 chromosomes. So our diploid number, our 2N number is 46. We get half of our chromosomes from our mom, and we get half of our chromosomes from our dad. So 23 from mom, 23 from dad. That's how we get our 46. And since we're getting half of our genetic information from our mom and half of our genetic information from our dad, that's why we are unique, yet a combination of both parents. Now, the chromosome number of an organism is unique. Different organisms have different chromosome numbers. And there's just a few examples on the bottom of this slide. So we said that we have 46, rice has 24, roundworm has 4, chimpanzee has 48, dog has 78. So what you need to understand is that chromosome number is unique for organisms, but having more or less chromosomes does not make you any more advanced or less advanced as a species. It's just how your DNA is packaged into these small structures. There are 3 main reasons why the cells go through the cell cycle. And the first is so that the cell stays small. This is for 2 reasons. One is to prevent something called DNA overload. DNA overload is when the cell becomes so large that the DNA no longer can control it. And so the cell will need to divide to stay small so that the DNA has control over the cell. And then thinking back to our last unit, small cells will allow for the most efficient exchange of materials because small cells will have a large surface area to volume ratio. So think back to the auger block activity that you had been working on in our last unit and how the small cell, the small cube, ended up losing all of its pink because diffusion was more efficient in a short amount of time. So that cell got whatever it needed or was able to get rid of wastes that it didn't need in a more efficient manner than that large cell, that large agar cube, which still had a lot of pink in the middle. Second reason for the cell cycle is for growth of the organism. So we start as a tiny infant, we grow to an adult, and our bodies are able to do that because our cells are dividing and making more cells. And then repair of damage or injury. So if we have damage to a certain tissue or organ, the cells are usually able to repair that damage or create new cells to repair that injury. There are specific stages of the cell cycle. The 2 main stages are interphase and the M or mitotic phase. And then those main stages can be broken down even more. Interphase has 3 of its own stages, G1, S, and G2. Interphase is most of the cell's life. The mitotic or the M phase has mitosis, which has prophase, metaphase, anaphase, telophase, and then cytokinesis. And the M phase is when the cell will actually divide, and that's a much shorter amount of time than when the cell is in interphase. There's some essential vocabulary that we need to understand before we hop into the different stages of the cell cycle. So DNA is something we've talked about before. We know that DNA is the genetic material of organisms. DNA is spread out during most of the cell's life as chromatin, another term that we talked about earlier. Chromatin is the form of which DNA is in most of the cell's life. It's when the DNA is all spread out and tangled. It's a big mass of DNA and proteins. It's like a big mess of string. It kind of looks like spaghetti inside the nucleus. Chromosomes are made of condensed chromatin. So if you look at the animation over on the right, you can see that the chromatin will start to coil and condense, and all of a sudden we can see these chromosomes. The only time we're able to see chromosomes is during cell division, only during the M phase of the cell cycle, because most of the time the DNA is all spread out as chromatin, and those thin strands of chromatin are not visible to us. But once they condense into the chromosomes during the M phase, then we're actually able to see them. If chromosomes weren't spread out as chromatin, this is what the chromosomes would look like most of the cell's life during interphase, kind of like that one narrow rod. However, we never see the chromosomes in that form, and that's because the DNA of the chromosome would actually be spread out and tangled as chromatin. So if it were to condense, we'd see it that way, but it does not condense, so we do not see the chromosomes in that form. After the chromatin condenses, the DNA forms the chromosomes that we normally see, that X shape of a chromosome. When we see the chromosomes in this X shape, what we are actually looking at are copied or replicated chromosomes, where that one rod, which we weren't able to see before, during the S phase of the cell cycle actually copies itself. So when the chromatin condenses during the M phase, now we're actually seeing 2 of those rod-like structures connected in the center. So when we see the X's, each half of the X is actually a copy. Each half is exactly identical. So those 2 halves of that chromosome are called chromatids, each identical half of a copied chromosome. These are often called sister chromatids. And remember, they're exactly the same. They're a copy of each other. The centromere is that, in the diagram, that yellow circle that's kind of in the center of our chromosome. This is the point of attachment of sister chromatids. So this is what's holding them together. Think of it like a rubber band that's holding those 2 individual rods together. If the centromere is split, the chromatids will separate from each other. So again, if we're thinking of that centromere like a rubber band, if that rubber band were to break, those 2 chromatids will separate from each other. So here's just a diagram that's showing an overall chromosome. The centromere would be in the center, and we've got the chromatids on each side of our X-shaped chromosome. A gene is a portion of a chromosome which controls a specific protein and controls a specific trait. When we get into genetics after a few weeks, we'll talk more about this, but our genes tell our body what proteins to make, and those proteins give us all of our physical traits. Now, since the chromatids are identical, the genes on the chromatids are also identical. So if you look at this diagram of our chromosome, and you see up here, we've got this gene, the one on this side would be identical to one on this side. But if we look at the bottom of the chromosome under the centromere, this down here would actually be a different gene. Each gene on this side and this side are identical, but because they're in a different location on the same side, all of these genes are going to be different, but then they'd be copied also on this side. So now the specific stages of the cell cycle. So interphase is the longest phase of the cell's life. This is where the cell is going to spend most of its time. During interphase, the DNA is spread out in that tangled mass of chromatin. And interphase has 3 stages of its own. The G1 stage is when the cell's doing all of its normal activities. It's doing what that cell's supposed to do. If it's a skin cell, it's doing the job of a skin cell. If it's a stomach cell, it's doing the job of a stomach cell. And during this time, the cell's actually physically growing. It's getting larger. The next stage is the S phase, and S stands for synthesis. This is when the DNA is copied or replicated. And we call that synthesizing DNA, thus the S. And then in G2, cells get ready for mitosis. It's going to get ready to go into that next stage. And so it's making sure that all the organelles are ready and that everything is set to be able to go through a division, which would be coming up in the next stage. So once we leave interphase, then we enter mitosis. And the first stage of mitosis is prophase. Prophase is when those condensed chromosomes now become visible. We can see them as their rod-shaped X form. The nuclear membrane begins to disintegrate. So the nucleus is going to disappear, which means the chromosomes are going to be able to just float in the cytoplasm. And at this point, the centrioles become active. The centrioles, if you remember from our cells unit, they help animal cells divide. And so this is when they're going to start doing their work. They actually start growing something called the spindle. The spindle is a form of the cytoskeleton, and it's going to help our cell to divide. Next stage is metaphase. Think of metaphase as middle. M for metaphase, M for middle. In metaphase, the chromosomes line up single file in the middle of the cell. The middle of the cell is called the equatorial plate. Think of equator. Or the middle of the cell can also be called the metaphase plate. When those chromosomes are lined up in the middle of the cell, the spindle will attach to the centromeres. So on each side, the spindle that was growing from those centrioles are going to grab onto that middle centromere. In anaphase, the spindle separates, and the chromatids are going to split at the centromere. So that spindle is going to pull the centromere apart. There's no longer a connection between those chromatids, and those chromatids are going to start to be pulled by the spindle to opposite sides of the cell. Now remember, each chromatid of a chromosome was identical. So now that they're broken apart, these chromatids on this side have exactly the same information as the chromatids on the other side. Okay? So even though our cell is going to split, the information going to each side is going to be identical. In telophase, the chromosomes gather together at opposite ends of the cell, and the nuclear membranes begin to reform around those chromosomes. And then in cytokinesis, the cell splits into 2 cells that are identical. The chromosomes spread out again as chromatin, so now we can no longer see that. And those two cells will start the cycle all over again. They will enter G1 of interphase and continue the cell cycle once more. So again, just a reminder of the stages. We've got interphase, which has G1S and G2. We have mitosis, which has prophase, metaphase, anaphase, and telophase, and then finally cytokinesis, the cell splits. And then those two daughter cells will go back into interphase and start the cycle again. So this animation is just quickly going through those stages of mitosis where we can see what's happening at each stage in the cell. These animations are showing you what's happening at those different stages as well. So you can see what's happening during early prophase, later in prophase, metaphase, anaphase, early telophase, late telophase, and cytokinesis. Now keep in mind that this is a cycle. And so it's not like the cell just automatically goes from prophase and boom, it's in metaphase. It's a gradual cycle. And that's why on these images, we've got an early and a late on some of the stages. Again, this is just some more animations that are showing the different stages of the cell cycle and what's happening. So just another way to visualize it. The average cell cycle time for human cells is approximately 20 hours. So a typical cell will go through all of these stages in about 20 hours. Now, some cells do this much more quickly, especially if there is repair that needs to be done. Some cells do this much more slowly, or maybe not even at all. And we'll talk more about that a little later. So this is an interactive mitosis activity that will walk you through the stages using both an animation and actual microscopic footage of the cell doing each of these stages. If you have not gone through this interactive mitosis on your own, be sure to go back into the notes slideshow that you copied and go through this. It'll be really helpful for you as you're learning these stages. So we talked about these major stages of the cell cycle, but there's another stage called G0. It's G0, it's not GO. So G0 is a non-dividing phase of the cell cycle. So cells can actually hop out of the cell cycle, go into this G0 non-dividing resting phase where they're not actively preparing for division. Now, if the cell goes into G0, it still exists, it's still doing its job, it's still performing all of its specialized tasks. It's just not going to do any of those preparations to divide. G0, there are differing times for differing cells. Now, some cells will never enter G0, like your skin cells. Your skin cells are continually dividing. They never hop out of the cycle. They just keep going over and over and over again. Some may enter for days or months. For example, liver cells. Liver cells don't need to divide all the time. They only need to divide if there's old worn out cells that need to be replaced, or there's been an injury or some sort of damage. And so liver cells might be in G0 for days or months, and then all of a sudden we need new liver cells, so they'll hop back into the cycle, do what they need to, and once enough cells are produced, they'll hop back out and go back into G0. And some cells, exist in G0 their entire lifespan. We do have some cells that never divide, like your brain cells, your heart cells, your skeletal muscle cells. So these cells do not have the ability to create new cells or to repair. So that's why if there is an injury or damage to brain cells, heart cells, or muscle cells, that can be permanent because these cells don't have that same capability of repair like our other cells do. This is just comparing the chromosome number. Remember, mitosis is making identical cells. And so we're going to start with 46 chromosomes and we're going to end with 46 chromosomes. We're able to divide the cell but remain with the same number of chromosomes because remember in the S phase, we copied everything. And so we've got an extra copy that we can give to a dividing cell. This again is also just showing how the chromosome number is going to be there as we go through mitosis. And it's using both the 2N designation, and it's also then turning that into numbers. Now you can see we start like on this side with 46. And then when we go down to this next cell, it says 46, and then there's a 46 in red. This is representing when we have all of our chromosomes copy, okay? And so then when they split, that's how we end up with 46 in each cell. So in summary, mitosis starts with one parent cell. It results in two daughter cells. The daughter cells are identical to each other and they're identical to the parent cell, and the chromosome number stays constant. Here is a review video on mitosis.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So, one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So, you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA, carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide. and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So, if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So, ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So, 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there. And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends, kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for two because you can really see in this step that the end goal is going to be two cells. And in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into two cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So, why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis.
This, once again, is just going through the chromosome number in mitosis, how we can start and end with 46 chromosomes. And that's because of that copying that happens during the S stage of interphase. This animation is a really good representation of the stages of mitosis. Now, animal and plant cells both do mitosis, but there's a few differences. And we've kind of alluded to some of these differences back in our cells unit. So, in terms of animals, animals have centrioles, and the centrioles are what produces the spindle fibers. Animal cells also form something called a cleavage furrow when the cell is starting to divide. The cleavage furrow looks like the cell is pinching in the middle. And we see that during telophase and then into cytokinesis when the cell is dividing. Plant mitosis, on the other hand, plants do not have centrioles, but they're still able to produce the spindle. The spindle just grows directly from the cytoskeleton rather than having that special structure to grow the spindle from. Another thing that happens is that we're not going to see any pinching, and that's because plant cells have a cell wall, and that's much too rigid for the cells to pinch. So instead, a structure called the cell plate is going to form in the center of the cell during telophase and cytokinesis. And this cell plate eventually becomes the cell wall, dividing those two new cells. So this is just a side-by-side view of each of the stages as an animal cell and a plant cell both go through mitosis. Now, for this process to happen correctly, there has to be some control. There has to be some regulation on what the cells are actually doing. So there are some cells that divide throughout the life of the organism. Like we mentioned before, our skin cells never take a break from division. They're constantly growing and dividing. Some cells divide only when given special signals, like our blood cells, like before we mentioned the liver cells that don't need to divide all the time, only when they're given a signal. And some cells never divide once they're formed. And we mentioned this before, like our nerve cells, our muscle cells, our heart cells. They're not going to go through this division cycle. So there are specific points within the cell cycle that things are checked and monitored to make sure things are going correctly. First, we have internal regulators. Internal regulators respond to events inside the cell. So it's going to monitor what's going on inside the cell to determine if the cell can proceed or not proceed with the cell cycle. This is mainly controlled by cyclins. Cyclins are proteins that regulate the cell cycle by helping the cell transition out of various checkpoints. So these proteins are going to make sure that when the cell goes from one phase to the next, that it's appropriately moving. So there are different cyclins that control different parts of the cell cycle. They're going to be monitoring different stages and allowing the cell to move or not move, depending on the job of that cycle. There are also external regulators. External regulators respond to events that are outside the cell. And external regulators are growth factors. Growth factors have the ability to stimulate cell growth. This would be like growth of an embryo, or when your cells are differentiating and getting their specific jobs, or in wound healing. So those are times when the cell needs to grow faster. Growth factors can also inhibit cell growth. And this has a lot to do with surface molecules that cells will stop dividing when they're in direct contact because the cells realize that there's enough cells there. And there's been experiments done that show this, and you can see this on the bottom, that if we have a petri dish full of cells and they're all touching, so that's going to inhibit or slow their growth. But if a strip of cells are removed from that petri dish, then that's going to cause the cells to stimulate their growth because they're no longer touching and crowded. And once they do refill that petri dish, then their growth will once again be slowed or inhibited. Now, the cell cycle has specific cell checkpoints. And these are stages where the cell uses both internal and external cues to determine to move forward or not to move forward. So the G1 checkpoint happens near the end of the G1 stage of interphase, and that checks to make sure that the cell is large and healthy. We want a large, healthy cell to move on. Now, if a cell does not fit the criteria for that checkpoint, there's a couple of options for that cell. One, the cell could kind of stay there, and if it's not large enough, wait till it grows a little bit more. Or if it's not healthy, maybe it can be repaired and get fixed before it moves on. The other option, if that cell is beyond repair, beyond healing, then the body can actually destroy that cell. And that's called apoptosis, when a cell basically commits suicide so that we don't have those damaged cells continuing to be reproduced. So if the cell is large and healthy enough and it makes it past the G1 checkpoint, it's going to go into the S phase, it's going to go into the G2 phase. But before the end of the G2 phase, we have the G2 checkpoint. And that's going to make sure that the DNA that was copied in the S phase was copied correctly. Again, if the DNA wasn't copied correctly, the cell's not going to be able to be moved on. And either the cell needs to heal that or it needs to get rid of that cell. So if it makes it past the G2 checkpoint, then we go into mitosis. And there's also a mitosis checkpoint before we get to cytokinesis. And that's going to make sure that the cells lined up correctly in metaphase, the chromatids were separated correctly in anaphase, because we want to make sure that those cells that are going to be produced after cytokinesis are going to have the right number of chromosomes. So those are the checkpoints that help to ensure that our cells are healthy as it moves through the cell cycle. This is another interactive activity that if you have not done this on the cell cycle and mitosis slideshow, you should go back and do that because it goes through those different stages we were talking about. It talks about the checkpoint, the G1 checkpoint, the G2 checkpoint, the mitosis checkpoint, and this also talks a little bit about that G0 stage, non-dividing resting stage for cells. Now, cancer, we've all heard of cancer. Cancer is when those cell regulators stop working. And when that happens, the cells will reproduce with no control. Usually, what ends up happening is that the cells reproduce much too quickly. And this can form tumors. This can form masses of cells that do not belong in that area. It can produce cells that aren't mature, that aren't able to do the job that they're supposed to do. Normal cells should be small, uniformly shaped with nuclei, a lot of cytoplasm. They should all be about the same size and shape. They should be arranged into tissues. They might have special cell structures, and they're not going to be dividing super rapidly. On the other hand, cancer cells sometimes are large, have kind of weird, odd shaped nuclei, not very much cytoplasm. They can really vary in shape and size, a lot of disorganization. They might not be able to do the jobs they're supposed to do, and they often divide much too rapidly. Now, there's two kinds of tumors. Now, remember, cancer is abnormal cell growth. Now, a benign tumor, a lot of time that's called non-cancerous, but it technically is a form of cancer because you have a group of cells that don't belong there. But when it's benign, the tumor is stationary. It's not going anywhere. And oftentimes, if that benign tumor is removed, everything's good. On the other hand, malignant tumors are usually what we associate with cancer, the disease. And a malignant tumor is a tumor that has the ability to metastasize. What that means is that it has the ability to break off from where it began and actually spread to other sites. And that's what happens when cancer spreads throughout the body. Malignant tumors are much more difficult to treat because they do have that ability to spread. So this is just comparing normal cell division with cancer cell division at a much more rapid pace and ending up resulting in tumors. Here's a quick overview of the cell cycle and cancer.
It is likely that you've heard the term cancer before. We have had family members that have battled cancer before. It is definitely a relevant topic for all of us. Cancer is in part due to cells that divide too frequently. The cells are not regulated, they are uncontrolled. Cancer cells can have other problems too. They might not be able to communicate with other healthy cells. They may not be able to carry out normal cell functions. They may not securely anchor themselves like other cells do, which can make them more likely to travel somewhere else. Some cancer cells have the ability to secrete their own growth hormone that makes blood vessels divert over to those cancer cells and supply the cancer cell with nutrients, which can take nutrients away from healthy cells. Why do cancer cells become this way? Well, there's a lot of research in this area. With some cancers, there may be genetic links, making some cells more susceptible to having problems. These genetic factors might run in families. Exposure to toxins, radiation, excessive exposure to UV light, all of these can be risk factors for some cells to become cancerous. The uncontrolled growth that cancer cells have gives rise to more cells like them, which can develop into a tumor. Some tumors stay put, but some do not. Now, the good news is that scientists continue to develop better treatments, which include destroying the cancer cells with radiation or medications such as chemotherapy, which will target cells that divide frequently. Maybe someday you will be part of helping to meet the challenge of trying to eliminate cancer, because the fact remains that these cells are not participating in the cell cycle like they should.